So let's talk about the photosynthesis experiment for a moment. Most plants undergo photosynthesis for their energy needs. Photosynthesis takes place within the chloroplast. The way photosynthesis works is plants take light, carbon dioxide, and water, and they transform that into glucose and oxygen gas. So when scientists try to study the amount of photosynthesis that happens, they have to measure something. And so here there's a couple of different ways how you can go about this and what you can measure. One of the things you could measure, for example, to try to see if photosynthesis is happening, is you could measure the amount of oxygen gas produced on the right-hand side of the equation. The more oxygen gas, the more photosynthesis. What we're going to do, though, is we're going to go a slightly different route. We're going to measure the carbon dioxide. So carbon dioxide and water are substrates in this reaction. And so if you have this reaction going on, more and more carbon dioxide will be turned into glucose. So this is how our experiment works, and that's what makes it a little complicated. So we're going to have to think through that, but I'll try to keep you on track as we're going through this. Just to give you an idea where all of this is happening. So all of this is happening in the chloroplast. So what you've got here is you've got a picture of a leaf, and you've already seen a leaf. You've seen the wandering Jew, you've seen the stomata of the wandering Jew. The plant was purple and the stomata were green. It was beautiful. And the stomata is where the gas exchange happens for the plant. So that's where the oxygen leaves and where the CO2 comes in. You'll find those on the underside of leaves. Then the actual photosynthesis happens in the thylakoid membranes, in the thylakoid space of the chloroplast. So you may remember this image from a biology book. So this is the overall reaction of photosynthesis. There are two parts to it. There's the light reaction, which is the one that takes water and light and uses oxygen as the final electron acceptor to produce the energy that's necessary to take the CO2 and incorporate it into the sugars. The CO2 is incorporated in the Calvin cycle, and it is not a light-dependent reaction. It's actually a light-independent reaction. But this is where the CO2 is being removed from the solution. So in the photosynthesis lab, what we're going to do is we're going to measure photosynthesis indirectly. So we're going to look at the CO2 that disappears from the solution. And the way you can measure this is because the pH changes when CO2 disappears from the solution. When you have a solution and you mix it with CO2, the CO2 reacts with the water and forms carbonic acid, which has a low acidic pH. You already know that this is the case because all of your soft drinks are slightly acidic. So the bubbles in your Coke, that's actually all CO2. And the reason why Coke dissolves your teeth and all of that stuff is because it actually is slightly acidic. Now, in the living organism, it doesn't make a difference, but that's why that is. So when we remove the CO2 from the water, then it raises the pH back to basic. That means your flat Coke is not acidic anymore. Now, the way we measure the pH in this experiment is we use a pH indicator. A pH indicator is something that changes colors depending on the pH of the solution. And the pH indicator that we're using is a pH indicator called phenol red. phenol red changes color based on the pH of the solution. Acidic solutions have numbers from pH of 1, which is like your stomach acid, all the way to 6, and then 7 is neutral, and then 8 through 14 is what we call basic. Phenol red is orange. When it gets slightly acidic, it goes actually all the way to yellow, the more acidic it gets. And when it goes basic, it becomes a deeper red. So at neutral pH, phenol red is red. It goes to orange and yellow when it goes to the acidic range. And it remains red, it just becomes a darker red when it goes basic. So this is how the procedure is set up. The first thing you want to do is you want to label 4 test tubes orange and 2 test tubes red. The way we usually do it, we just take 4 test tubes that get an orange lid and 2 test tubes that get a red lid. You're going to make 2 sets that are identical. You're going to get a flask with phenol red, pour it in the 2 test tubes labeled red. That's going to be your negative control. That's what a non-CO2 phenol red looks like. Then you're going to take the flask and you add CO2 to it by blowing air into it with a pipette. When it turns a little bit orange, you take that and put it in the 4 test tubes labeled orange. Then you place the plant Elodea into 2 of the orange tube and put a green dot on them. Cap all the tubes. So you now have 2 sets of tubes, 2 red ones, 2 orange ones, and 2 orange ones with a red dot on the top. And you're going to set one of these sets into the dark and one of these sets into the light. And this slide gives you a visual of this experimental setup. So you start out with a beaker filled with phenol red, put that in a tube, blow into the beaker to add CO2. That causes the formation of carbonic acid and turns the phenol red orange. Then you're going to take a sprig of Elodea, put it into one of the tubes, and that's one of your sets. And the second set, we're doing a second set just like that too. So here you see what our tubes would look like. On the top, you see the 2 sets, the way the solutions look. The basic is on the left, the acidic is on the right. So the acidic is the one that has the extra CO2 in it. And then on the bottom, you see the 2 racks. The rack on the left is going in the dark. The rack on the right is going into the light. And each of these racks has one of each type of tube in it. So a red-lidded one, a orange-lidded one, and one that has the orange lid with the green dot, which has the plant in it, and the CO2 enriched phenol red. All right, so here we are. We're going to put 3 of these tubes into close to a light source, close to the sunlight maybe. If we have sunlight, if not, then just a strong lamp will do. And 3 tubes on the bottom, they go in a drawer. So here we are. After 45 minutes, we're going to remove those tubes from the light and from the dark, and we're going to look at them. And what do you see? On the next page, you're going to be able to look at the data table results and record your results on the data table. Spend a couple of minutes looking at this slide and looking at the data table to see what you think. On page 17 of your handout is a data table that looks sort of like this. So you have the treatment on the left, dark versus light, then the solution at the start, orange, red, or orange with a green dot, that's the plant one. And then after 45 minutes, you need to be looking at what your plants look like. On this slide, you see an overview of the whole experiment. So on the left, you see the pictures at the start of the experiment. Top, you have the light. Bottom, you have the dark. And on the right-hand side, you see what it looks like after 45 minutes. On page 17, you're now asked to fill out a couple of questions. So first, you look at the tubes that don't have any color that don't have any plant in them. Did they change color? So can phenol red change color on its own? The second question asks you to look at the tubes that do have a plant in them. Did any of those change color? Can you explain why? So spend a couple of minutes looking at both these tubes and the handout, and try to see if you can answer the questions in the handout. I realize that this experiment is relatively difficult to understand, and it's relatively hard to keep all the things in mind that you're having to keep in mind all at the same time. So I'm putting this together to remind you again what the reaction is that we're looking at. So what we're looking at is the reaction of photosynthesis that takes carbon dioxide and water and light energy and turns it into glucose and oxygen gas. And so we're looking at the activity of photosynthesis by looking at the disappearance of carbon dioxide in the solution. So look at the start of the experiment. The red tube has very little CO2 in it because you added CO2 by bubbling it into the orange tubes. So the orange tubes have more CO2 in it. So that was the startup in both the light and the dark reaction. Now after 45 minutes, you still have very little CO2 in the red tube. And you still have the same more CO2 in the orange tube. So in the dark reaction, the tube is still orange. What does it tell you about the amount of CO2? And what does that tell you about the amount of photosynthesis that was going on there? In the light condition, you have a red tube, so it changed. So something changed about the amount of CO2 there. Is it more CO2 now, or is it less CO2? And what does that tell you about what was going on with regards to photosynthesis?